I'm also going to introduce to you Dr. John Seebeck. He's one of our long-standing UBRP mentors. In fact, I think you may have broken the record for the number of UBRP students mentored, close to 40. A lot over the years. And he's going to give the introduction to our speaker today. Experienced.
Well, it's my pleasure today to be able to introduce the speaker for today. That's Dr. Eric Walk. He is the Chief Medical and Science Officer, or Scientific Officer rather, for Roche Diagnostics, which is part of Ventana Medical Systems. They're up here in Oro Valley. He's also the Senior VP. And as part of what he does, he oversees a group of over 130 employees, and they're responsible for overseeing clinical trials that can lead to clearance of some of their materials at the FDA. He also does some exciting work in translational research, developing novel new medical products. And so today he's going to talk to you about some of the research that's being done up there at Ventana, as well as opportunities for you to get involved. Dr. Walker.
Thank you very much, Dr. Seebeck, for that very kind introduction, and welcome to all of you. I have to say to start that I'm incredibly impressed with the UBRP program, so congratulations for being selected into it. I was just speaking with Jennifer before this lecture, and she was telling me that about 20 or maybe even 30 UBRP grads have either worked or are working at our company in Oro Valley at Ventana Medical Systems. And we're looking for ways to even build stronger ties to the UBRP program. In fact, one of your classmates, Ricky Lyra, can you feel here? No, Ricky?
No, this is the new group.
Anyway,
So.
one of your classmates that you'll soon meet, I'm sure, is doing an internship at our company, and I'll talk a little bit about the internship program during my talk. So as you can see from the title of my talk, what I'd love to talk to you today about really is my journey as a scientist and a physician, and how, even though I never, when I was your age sitting where you're sitting, I never, 0% thought that I would end up where I ended up. How did I get there? And why do I love it so much? That's really what I want to tell you about today. And along the way, I'll talk about how you can pursue a medical and or a scientific career, how you can pursue those interests within industry, and do it legitimately. Pursue real science, real medicine, solve real problems, and actually help a lot of people by doing it. I know we have time at the end, I think 15 minutes for questions, but I actually prefer an interactive session. So if I'm presenting on something that you have a question about, feel free to shout it out or raise your hand or however you want to do it, and we can also do questions at the end. Just so I get a better sense for all of you, how many of you are freshmen? Sophomores? Juniors? And seniors? Great. Okay, that's all. Thank you. So this is the agenda. I've split my talk into 4 big parts. First, I want to tell you about my journey, how via serendipity I ended up as a physician in the pharmaceutical and diagnostic industry. Then I want to tell you a bit about the company, Ventana, and my specific role in the company, what I do. I'll then move into describing in general terms how one could have a career in industry through science and medicine. And then I'll finish up with what I find to be the most interesting part. I want to tell you about some of the research that we do at Ventana Medical and how some of that research has led to products that are actually helping patients around the world today. Hopefully you'll find that enlightening. So I'll start with my journey. This is more or less my roadmap. When I was your age, I was an undergraduate student. I thought I was pre-med. I thought I was going to go to medical school. I thought I was going to treat patients, and I was just narrowly focused on that pathway. And as you can see by looking at some of the changes in my career, I didn't end up doing that at all. I did practice pathology for a brief time, but went fairly quickly into the pharmaceutical industry, first with Novartis, and then later eventually made my way to Roche through Ventana. Ventana was acquired by Roche in 2008. So it has been a very different journey than I expected. And you'll see that the subtitle here I have, Follow Your Bliss. How many people have heard of this term, follow your bliss? few people. Okay, so this is a term coined by a fascinating individual named Joseph Campbell, who was really a mythologist. He wrote many books, but this is one of his most well-known books, The Hero with 1,000 Faces. And he really coined this term, and if you read that passage, maybe I'll read it. If you do follow your bliss and put yourself on a kind of track that has been there all the while, waiting for you, and the life that you ought to be living is the one you are living, follow your bliss and don't be afraid, and doors will open where you didn't know they were going to be. So have you ever had that feeling when your mind is telling you to do one thing, but your heart, your soul, your spirit is telling you to do something else? Have you ever experienced that? So that's what Joseph Campbell is talking about. And what he advises you to do, and what I found very useful in my career, is to follow your heart and your spirit versus maybe what your mind is telling you to do. And find that pursuit, whatever it is, whether it's biology or research or diagnostics or cancer research, find that core pursuit that resonates with you so strongly and that you're naturally good at, that things come easy to you. That's what I have tried to do in my career. That's my, this advice came from my father, actually, and I found that it's been very helpful to me. And I attribute this simple philosophy to any success that I've had and the career path that I've followed. So follow your bliss. So a couple of years ago at a leadership retreat, we did an exercise called Journey Maps. And you're probably too young to do this yet. You can start your journey map, but maybe 10 years from now, I encourage you to maybe look at this again. And so what we did is we plotted out our life path and all the key decision points. And you can see, I'm not going to go into the details, but you can see, my path certainly wasn't a straight line. So when I was a kid or when I was in college, I thought I was going to be pursuing this path. My father was a gynecologist, so I thought I was going to be a gynecologist just like him. So I was going to go to medical school, then do a G1 residency, and then open a practice with him. And of course, that didn't happen in medical school. I decided I hate gynecology. I absolutely hate it with every cell in my body. And so the clock here pointed 3 o'clock, so I had a 3-hour heart-to-heart talk with my dad, telling him that I couldn't be partners with him in his GYN practice, that I really loved pathology, and I always had. So when I was a kid, my favorite toy was a microscope. And I would make my nose bleed so I could collect the blood to look at it under the microscope. Yeah, you're a kid, right? And then in medical school, pathology was the class that came to me the easiest. Whatever the assignment was, I was able to find the cell or the tissue or the structure first, and all my other classmates would come to me for help. And so this was the first sense that I got that pathology might be my bliss, but I was conflicted because that was what my heart was telling me to do, but my mind was telling me I need to be a gynecologist so I could practice with my father. So it all came to a head at this point in time when I had to make that decision. This happened to be third year of medical school when you have to make a decision as to what residency you're doing. So I made that tough choice, and that was a real fork in the road for me to away from gynecology towards pathology. So at this point in time, I thought, okay, I'm going to be a pathologist for the rest of my life. But then I got to pathology practice after residency, and I found it boring. I still love pathology, but the day in, day out practice of pathology wasn't at all what I thought it would be. It was not academic, it wasn't molecular, it was very boring to me. So then, through a headhunter, I found an opportunity at a pharmaceutical company, Novartis, in New Jersey, and loved that. So for that period of time, right around here, I thought, okay, I'm going to be Eric Walk, the pharmaceutical scientist, pathologist. That's going to be my thing. And it was my thing for about 4.5 years. Then I met Tom Grogan, who founded Ventana, and I'll tell you about Ventana in a second. And Tom, as a single person, made me change my path again. And he recruited me to Tucson. I started with Ventana in 2005 and haven't looked back. I've been there ever since. So at this point in time, who knows how the rest of my journey map will turn out, but at this point, I'm thinking this is my bliss. And I am Eric Walk, pathologist, who's a chief medical officer at a diagnostics company. So my message to you is, you're back here somewhere. Have a plan, have a career path, pursue your interests, but just realize that there's a pretty good chance that your map is going to be as branching or even more branching than mine is, and be open to those opportunities as they come along. So, what about Ventana Medical and my role? So I already talked about Dr. Grogan. Has anyone heard of Dr. Tom Grogan? A couple of people, great. So that's Dr. Grogan. Tom started as a professor here at the university in anatomic pathology. And back in the mid-'80s, he realized that as good as a pathologist as he was dependent on manual tests called immunohistochemistry. Has anyone heard of immunohistochemistry? Okay, a couple of people. Immunohistochemistry is essentially a fancy way to say looking at proteins on tissues, on glass slides. And back in the 80s, you had to manually pipette the reagents and bake these slides in ovens and take them out at specific times and titrate different reagents, all manually. About 30 steps, 2 days. It was a lot of work and highly variable because there's the human element. So Tom realized this, and his vision was to create an automatic machine that did that better than the human. And that's how Ventana started. I probably shouldn't tell this part at the U of A, but when Tom spun out Ventana from U of A, he was considered by the university as a Class 3 fellow, because that was against the bylaws of the university at the time. But Tom didn't let that stop him. He continued to do that and eventually got the bylaws amended. So now, as you well know, actually I believe the university is very much in favor of having companies spin out of the university. But you could say that Tom really was the pioneer who started that. Unfortunately, Tom was diagnosed with melanoma in 2015. But to his credit, he continued to pursue diagnostic innovations, and through those innovations, he helped cure himself through some of the tests that I'll tell you about. So it's a really amazing part of the story. A few statistics about the company. So we employ almost 1,400 employees at this point. We have over 12,000 of our instruments around the world in almost every major country in the world, and a menu portfolio of over 250 tests. So our company business model is, if you will, a razor blade model. So we sell testing instruments, and then we sell the tests that go on those instruments. So that's what Ventana does. So we're both, because of that, we're both a hardware engineering company as well as a life science company. And I'm going to talk more about the life science part of the company today than the engineering, but just so you know, we're both. We are, from a disease perspective, we are 100% cancer focused, which is my primary interest. As you know, cancer continues to be a worldwide health problem. How many people in the room here, by show of hands, have been affected by cancer? A friend, family member, or loved one? Yeah, so it's virtually everybody. So that shows you the need that's out there and why we all come to work every day. It's at least why I come to work, because improving the lives of all patients afflicted with cancer is a mission that really resonates very strongly with me, and I think that goes the same for all the people that work at Ventana. So we aim to achieve our mission through developing the highest quality, most innovative cancer tests out there on the planet. And most people don't realize that up in Oro Valley we have this company that's a worldwide leader in cancer testing, but that's exactly what we are. A little bit about my role at the company. So as Dr. Sivak mentioned, I'm the Chief Medical and Scientific Officer. I sit on the leadership team and report to the president of the company, Ann Costello. And it is my responsibility to run 3 groups, medical and scientific groups within the company. I'm really not going to go into the detail, but we have a group of pathologists because that's very important for a tissue diagnostics company. I will talk a little about medical affairs and what that is. And then we have a 3rd group that I won't talk about, which is called clinical science, which is more around the medicine and science of registering our products through the FDA and doing the clinical trials. So if you ask me what are my primary roles and responsibilities, the first I would say is leadership. And I would say if you're interested to pursue a career in academia or industry, I would advise you to do as much as you can in the way of leadership. It's not too early. You may think it's too early to develop leadership skills at your stage, but I would say not. That once you get either into academia or into industry, people need leaders. So you need, especially scientific leaders. So people who are credible from a medical and scientific perspective, but also can lead teams of people. When I say lead, what I really mean is inspire. So what leadership is to me is it's inspiring a group of people to achieve a certain mission. You saw what our mission is, and that's what leadership is. And so I provide this leadership to the medical, clinical, and scientific team at the company. Some other things I do, more in my chief medical and scientific officer role, I'm responsible for engaging with the external community. So I spend a lot of time speaking with people at the university. I speak to the FDA quite frequently. I speak to pathologists and oncologists around the world about collaborations and projects we can do. And locally, I don't know if anyone's heard of the Southern Arizona Leadership Council or BIOSA, but I work locally with a lot of biotechnology and industry advocacy groups trying to strengthen the ecosystem, if you will, for industry biotechnology in Southern Arizona. And I really enjoy that. And given our mission as a cancer company, of course, we need to interact with a lot of pharmaceutical companies, including Roche's own pharmaceutical company, Genentech. But we also work with all the major non-Roche pharmaceutical companies. As a physician, I also serve as a safety officer, if you will. So I was on a call with FDA just yesterday, talking about a product issue that we're having and answering questions that they had about how much the safety risk of that product issue has to patients out in the field. Thankfully, we don't think there's much of a safety issue this time, but if there were, we would need to take specific actions, and it's my responsibility to oversee that and really act as a bit of a medical conscience for the company, if you will. And last but certainly not least, it's actually my favorite role in the company is really to be part of the innovation engine for the company. So a company like Ventana, we don't want to be a me-too company. We don't want to just create products that our competitors are creating. We want to be innovative and cutting edge, and that requires interacting with academic experts like at the University of Arizona. It requires bringing in novel technology and really having some very creative ideas about how technology can help patients with cancer around the world. I'll talk a little bit about that through the examples that I'll show. Any questions about anything that I've said so far before I continue? You all with me? Good. Okay, so now I want to talk generally about how one could pursue a medical and science career within industry. And I really want to start by describing what industry is all about. So, and let me ask you, what do you think the primary goal of a healthcare company is? Just shout it in here. Or let me lead you a little bit. They want to create what? What does that healthcare company want to create? Yeah.
Technologies or techniques to be used in the medical field.
Yeah, exactly. Or to phrase it another way, a product, right? A product that's a technology or a technique. So that's exactly it. And it's as simple as that. So in the academic world, clearly the academic world is not focused on developing a product. The academic world, you could tell me, but one of the things the academic world is focused on is discovery. The industrial or commercial world is not so much focused on discovery. They're focused on developing a product. And because of that, we need to do research. And the research leads into product development, which can take years, and that leads into commercializing a product. And so this becomes a cycle, because the products that we commercialize and sell generate money, and that money then gets funneled back into research and development to develop new products. So this is how all companies work. So if you're not successful as a company, then your product development comes to a halt, and patient innovations also come to a halt. So some people grapple with, I didn't so much, but some people grapple with, well, how can I join a company? And can I still be ethical? Can I have scientific purity or medical purity by joining a company? For me, I feel that I completely have scientific and ethical purity because all the work that I do going into product development, generating products that help patients, generating funds to put back into research for other products, for me there's nothing better than that because the products that my team helps develop are helping millions of patients around the world. So that's how I think about this product development cycle. Of course, I'm leaving a lot of detail out. I'm not going to go into this, but just so you know, there's a lot of detail when one is an FDA-regulated company like ours. There are a lot of things that you have to do along the product development process, from the early concept to having a design goal, to figuring out exactly during feasibility what your design inputs are going to be. So what are the specifications that you want to meet? Then at the end of that process, which may include a clinical trial, you want to measure what did you create. Did the specifications of the product that you created meet the specifications during that input phase? Then you have to come to a point where you have to decide, is the product good enough to launch? Will it be successful? So that's a specific milestone. And then after the launch of the product, we typically look back a year or two later to see if the product did actually do what we intended it to do. And you see a lot of detail along the way that I'm skipping. But suffice it to say, this is why we have 1,400 people in the company developing products. So at a very high level, now to get to the science and the medicine, how does science and medicine feed into this? Well, it's critical. So without science and medicine, there would be no Ventana, and there would be virtually no healthcare company. So, as I just said, research and development involves a lot of different activities and processes, but the input to the R&D process itself comes from a realization of unmet medical needs and an assessment of different technologies. And without these, there is no product to develop. And so we spend a lot of time in the company in both areas trying to identify what are the most compelling unmet needs for different patients with cancer, and what are the most innovative, creative technologies that we can bring and pair with those unmet medical needs that will create a product. And after years of research and development and a clinical trial going through the FDA, hopefully a product will come out at the end. So that's the overall process. Now you may say, well, what are the scientific skills that are required? And this is really an incomplete list of some of the skills that in Ventana are absolutely necessary for developing these products. And I've tried to limit this to the scientific skills, the non-engineering skills, and so I won't go through them all, but we critically depend on biologists, molecular biologists, chemists, as well as people who analyze our experiments, biomedical engineers, pathologists, people who know something about antibody development and purification. You know, for us, these are absolutely critical skill sets. So if any of these are connected to things that you're pursuing, then know that you have a skill set that can be applied to your industry. And this, again, is a list from our point of view and perspective at Ventana. This list would be slightly different if you spoke to a different diagnostic company or a pharmaceutical company, but I think a lot of the lists would overlap as well. On the medical side, there's also a lot of important skills that are required, and the process here is about understanding what are the unmet medical needs out there that can be improved through a medical device or a diagnostic. So we spend a lot of time looking at different diseases and understanding during the screening phase or the diagnosis phase, during the treatment phase or the follow-up phase, the monitoring phase, where are these needs? So what are patients not having done to them today through a test or through a treatment where we could develop something new to help them and help their physicians make a clinical decision that would end up benefiting their life. I'm not going to go into the details, but you can see the detail to which we do this. So this is for breast cancer, lung cancer, and prostate cancer, but we literally map out the diagnostic and treatment process for each of these patients, and then color code each of the decision points in yellow, green, or red. Green meaning there is an opportunity. Red meaning either there's no opportunity or the opportunity has already been met by an existing product. And yellow meaning something in between. So this way we can prioritize the unmet medical needs that we want to pursue and then develop our products based on these other medical needs. I mentioned before about medical affairs, and has anyone heard that term before, medical affairs? So medical affairs is that part of medicine and science that's applied to products after they get launched. And so, as you can imagine, you can develop a product that's great, you can launch it in a fantastic way, but if you don't provide, especially with innovative products, new products that no one knows anything about, if you don't provide your customers with educational materials or training materials on why that product is good or how it's used, you can imagine that if you don't do that, that the product won't be very successful. So this is essentially what medical affairs is about. It's about providing the data, the knowledge, the publications that provide education and training to our customers out in the field so they know what our products are, how they compare to other products, and how the products can ultimately help patients. And so we have teams of physicians and scientists actually working, not during product development, but during this post-launch phase to help patients and our customers, who tend to be physicians or laboratorians, understand what our products do. So it's another way that you can have a career as a scientist or as a physician in industry, but not in a product development perspective, but more as an enabler of product adoption. I want to talk a little bit about the internship program that we have at the company. So we've had this relationship with the university, so we've had interns come from the university, I think, for over 10 years. This year, we have 10 interns coming from the U of A. You can see the departments that they're going into. We're very proud of this. And has anyone here been part of the internship program or know someone who's participated in the Ventana program? But you know someone? Yeah. Yeah, fantastic. And so what they do is they spend a number of weeks at the company and they get paired with a mentor in one of our scientific departments, and at the end they present a poster at a poster fair, and I go every year, and I'm always impressed with what the students are able to do in a relatively short time. We hope that some of the interns will come back to us as employees, and these are some of the career paths that would be available to interns or people coming to us right out of college. And some of these, would you have a question?
Yes, this is whenever you're going to.
No, please.
So are these undergraduate students or are these graduate?
These are undergraduate students participating in our internship program. And so we're showing some of the career options that would be available to undergraduates once they would join the company. And some of these would require some years of experience afterwards, but these are the general career paths. And we actually have two examples. I don't know, does anyone here know either of these students? Andrew or Andrea? No, maybe there's some. Jennifer, do you recognize them?
I would if I had their last names.
Okay. So these are two examples of interns that came from U of A who ended up working at Ventana. So in the case of Andrew, he's currently a scientist too. He actually did 3 consecutive internships within Roche, and we hired him in 2017. And Andrea did 1 internship. Initially was a contractor for us, and then got converted to a full-time employee. And actually works in one of the departments that I used to oversee in clinical operations as a clinical research associate. So there's two examples from the University of Arizona of pathways that went through the internship program and ended up as employees of our company. These are some of the general types of roles that undergraduates who newly graduate and join our company could come into. You can see the years of experience there. So typically you would start as a research associate and through years of experience work your way up to principal scientists. We even have senior scientific fellows that come after that for people who have really done outstanding scientific work. But just to show you that within industry you can have a career path as a scientist and have a lifelong career if you chose within industry. And we have a similar pathway for physicians that I'm not showing. That's one group that I run at the company, the pathology group. And also we have many pathologists who've been there right out of residency and intend to spend their entire careers within the industry. So I'm going to spend the rest of the time that I have, and is it till 4:30? Yeah, so roughly half an hour, going through what I feel is the most interesting part of what I do, and that's the actual science in medicine driving product development. So depending on time, I'll go through either one of these examples or both of them. And what I intend to do here is to show you two examples of products that started as scientific concepts and how they were developed and made their way to benefiting patients around the world today. The first is a product that's called synthetic histology, and this is involving a biomarker called P16 that I'll describe. And this is a product that was approved a couple of years ago, and it's on the market now. But I'll describe to you the clinical scenario. So this is cervical cancer, and as you may know, cervical cancer, unfortunately, despite the fact that it's completely preventable, remains a tragic health problem today worldwide. As you can see on the slide, there are over 500,000 cases diagnosed per year, with a shocking 266,000 deaths per year. Fortunately, most of those deaths are not in the U.S., but still, to have that many women dying of a preventable disease is really unacceptable in 2018. And you can see the rest of the statistics there. It's the fourth most prevalent female cancer, etc. I'm sure you know that HPV, the virus, is the cause of cervical cancer. And this is it's a graphic to show you how cervical cancer progresses from an HPV infection through a phase that we call pre-cancer to ultimately invasive cervical cancer. So the difference between this pre-cancerous phase and full cancer is a tumor's ability to metastasize. So pre-cancers can't metastasize. Cancers can. So when a cancer metastasizes and spreads to the brain or the liver or the bone marrow, that's how patients die. So the opportunity is to catch cervical cancer in this fairly long pre-cancerous phase. And so what's interesting about this disease is it's caused by HPV, but not all HPV infections lead to cervical pre-cancer. That's very important to understand. So just because a woman is HPV positive doesn't mean they're going to have pre-cancer and let alone cancer. The vast majority, actually, of HPV infections, 70–90%, clear and completely go away. For reasons we don't completely understand, some of these infections lead to transformation in this precancerous phase. So I've got to teach you some terminology here. So the real term for this precancerous phase is cervical intraepithelial neoplasia, or SIN. And there's 3 different grades of SIN: SIN 1, 2, and 3, with 3 being the worst. And so this progression to cancer can take anywhere from 20 to 30 years. So it's very, very slow, which is why there's ample opportunity to catch the disease in the precancerous phase, the SIN phase, and treat it before it ever gets to the cancerous phase. So that's why cervical cancer screening is so important. Any questions? Yeah.
would signals for this type of development be caught during like pap smears or regular occasions, or does it have to be a specific you go in and get it tested for this?
Yeah, absolutely. So that's what I'm going to be talking about. So it was a perfect setup. So the historic method of cervical cancer screening is exactly what you said, is the pap smear, the pap stain. And what that is, I don't have that slide, but the pap smear is collecting cervical cells, putting them on a slide, staining them with a Papa-Nicolau stain, which Dr. Papa-Nicolau developed almost 100 years ago, and then a pathologist looking at the slide and determining if those cells are dangerous or not. The problem with that is, unfortunately, 50% of diseases missed using that particular technique. So using this technology, or a version of this technology actually that I'm not talking about today, we aim to improve that rate of detection to 90% versus 50%. But currently that is the standard of care. We're looking to improve upon that, however. So just to follow that path, so the way women get screened is through the pap smear. Any slide that looks suspicious then is triaged to a cervical biopsy. And then, so the biopsy, rather than scraping cells from the cervix, the biopsy takes a core of tissue from the cervix. That is the definitive way to prove that a woman does or doesn't have the precancer. So you understand, so it's the screening phase and then the diagnostic phase. So, and actually that's exactly what this slide is about. So the pap smear is this screening phase. So you look at the black dots. So the black dots are suspicious women. And what we're trying to do is narrow down which one, in this example, which one woman actually has cervical precancer. But to get there, we need to go through this triage phase, which currently is done through the pap stain and some other technologies. We're trying to improve that as well. So the focus of my talk today is actually, I want to tell you about a product that we're developing in this diagnose phase. So this is the phase where confirmation of the precancer occurs through a pathologist. And that's typically done with a pathologist looking at a slide stained with hematoxylin and eosin, or H&E. Has anyone heard of H&E? That'd be really impressive if someone's heard of H&E. Okay, a couple of people that have heard of H&E. So you're looking at H&E. So H&E is a purple and a pink dye. Again, hundreds of years old. So the purple stains DNA, so that's the nuclei. The pink stains protein, so that's the stroma. And this is just how pathologists look at cells and tissue and make diagnoses. So this is what the normal cervix looks like. The normal cervix is supposed to have a basal layer of round plump cells that flatten out as you get to the top. So these cells divide and they push cells up, and then eventually those cells flatten out and then eventually lose their nucleus and swell off. So this is what normal looks like. This is what precancer looks like. And there's two flavors of precancer, low grade and high grade. Low grade cells, or low grade biopsies look like this, where you have, if I can convince you, the big cells at the bottom now are rising a little higher. And what's happening here is HPV has caused these cells to divide faster than they should. So they're dividing and pushing those bigger cells up higher. And if you look over here, that's in the extreme. So now the whole thickness of the cervix is those big cells. So this is CIN3, or high-grade cervical precancer. This is low-grade precancer, and that's normal. So remember these images, because I'm going to quiz you at the end on your knowledge, including you, Jennifer. Okay, so that looks pretty clear, right? Normal versus low-grade versus high-grade. But of course, we've got to make it more complicated, or biology has to make it more complicated. And the complication is that you have mimickers of precancer that are completely benign. So you have things like metaplasia, transitional cell metaplasia, atrophy. So these are all benign things, but they look like the precancer, right? Okay, so I'm going to teach you in about 60 seconds to tell the difference, and then I'm going to quiz you at the end. Completely unfair, but I'm still going to do it. So this is it's called squamous metaplasia. These cells look plump, but if I can convince you, the cytoplasm is very pink and round. And the real clue is that you've got a gland here. So this metaplasia tends to occur around glands. And actually, what metaplasia means is transition of one cell type to another. So here, these cells used to look like this, glandular cells, but now they've transitioned, they've metaplased into squamous cells. And so that's what this is. Completely benign. Transitional cell metaplasia. So believe it or not, these cervical cell types can turn into a cell type that you would see in the bladder. That's what transitional cells are. The clue here is, I don't know if you can see in the back, do any of you see a nuclear groove there in that cell, that line? That's the clue. So when you see that line, that means it's transitional cell metaplasia, not cervical precancer. And finally, atrophy. So this tends to occur in older women, and it's what it sounds like. It's a flattening of the epithelium, and so it looks like you've got a lot of those plump cells, but it's just that the top layers are gone. So the clue is when you see something flat like that. So got it? Everyone's now an expert in low grade and high grade CIN and squamous metaplasia, transitional metaplasia, and atrophy? Good. Okay, so if you thought that was confusing, you're not alone. So, and this is a little scary. So practicing pathologists out there mess this up all the time. So what you're looking at is data comparing expert pathologists to community pathologists. So I'm talking about pathologists who only do GYN pathology versus pathologists who do it sometimes, at St. Elsewhere somewhere, along with head and neck pathology and liver pathology and everything else. So I won't go into the details, but basically they mess up these critical CIN categories up to 50% of the time. That's not good. They're overcalling, they're undercalling, they're not accurate. So that is an unmet medical need. That's a problem, right? That's a misdiagnosis that could impact patient care, because if the pathologist is saying that the woman has precancer when they don't, then that woman's going to get a biopsy or a resection of her cervix unnecessarily, and that's no good, right? And then the other way around, if they undercall cervical precancer and the woman doesn't get her disease removed, that's almost worse. So this is an unmet medical need. So what's the solution? The solution is to remove the subjectivity. So we did a lot of analysis and research to see, is there a biomarker that we could apply to slides that would help pathologists be more accurate in diagnosing this form of cervical precancer? And we found one called P16. And so essentially P16 is expressed during this transformation phase. So remember I told you the HPV cells get infected, the patient gets infected with HPV, but not all the time does it progress to cervical precancer. Well, this P16 biomarker is only positive when the transformation occurs. You might be interested in the biology behind it. So essentially, this P16 is under the control of a promoter that is typically repressed by a complex of E2F and phospho-RB. Phospho-RB, you may have heard of, is the retinoblastoma gene. It's a tumor suppressor. So when HPV infects the cells, HPV, one of its proteins is this oncoprotein called E7. So E7, what it does is it comes along, it displaces E2F and binds to phospho-RB, releasing this block. So then this promoter is essentially activated, and the P16 gene gets transcribed and expressed. And the expression of the P16 gene can be detected through our technology, immunohistochemistry, which I said before is a fancy way of looking, or saying, looking at proteins on slides. So the brown staining that you see there, that is our IHC test showing positivity for P16. So when you see brown in this test, it's positive for P16. So how can we use this test? So remember before I told you it was very subjective. The pathologist would look at the cervical biopsy, decide if they saw those grooves or if they saw enough cytoplasm, and decide if it's precancer or one of the mimickers. But with our test, they don't need to look at any of that. They just need to see, do they see brown staining? So here's a H&E slide that was initially thought to be benign. You run our test and you see brown staining. And so the P16 is positive, implying that the HPV transformation process occurred, implying that this is cervical precancer, not something benign. So that diagnosis was reversed. Here's another case. It looks like metaplasia. It really does look like metaplasia, even to me. But the test was positive. And so this diagnosis was reversed. Here's a case that was thought to be CIN2, so thought to be cervical precancer, but the test was negative. So this diagnosis was downgraded to benign. So you can see how this test can help correct some of the errors that might occur out there in the field, even with well-trained pathologists. Another example, the pathologist didn't know what this was. Is this metaplasia, which is benign, or is it CIN2? Test is negative, they diagnosed it as benign metaplasia. Question?
Yeah, that's kind of like a big question.
Yeah, sure.
In terms of like development or choosing a protein of interest to use as a biomarker, like how does a team go about finding what P16 is anyway?
It's a great question. And I wish it were an easy answer, but it's not. A lot of it is brute force, and a lot of it is trial and error. And like I said before, companies are not set up to do discovery. So I can't say that we have this massive discovery effort to look for this biomarker. Essentially what we do is we scan the horizon of academic discoveries. So we look at publications, we look at discoveries coming out of specific labs, we go to all the cancer conferences to see who's presenting on what. And so we came across this particular discovery that came out of the German Cancer Center. And actually a small company had developed out of it, and we acquired the company. That's how this one happened. But that's how academia and industry can work together, because academia is set up for discovery. Industry is set up to take those discoveries and turn them into products like this. So that's why it becomes a translational partnership. Does that answer your question? Yeah.
How high is like the accuracy for this test? Are there a lot of like false positives or false negatives?
Again, it's a fantastic question. If I could hold the response, I'll show you the data. So we did a study to look at exactly that, and I'll show you in a second. Great question. So this test can also be used to detect small pockets of precancer that a pathologist might miss. So here, there's a lot going on in this slide, and I would have looked right by this, but if you look at the test, you can see here, hiding amongst all this junk were clusters of precancerous cells. So this case actually was positive for precancer. So based on this information, and I'm glossing over a lot of it, but there were literally hundreds of articles from around the world, different academic groups, studying this P16 biomarker, which is important to your question about accuracy. So based on all of that data and the trial that we conducted, in 2012, the College of American Pathology, the American Society for Cervical Colposcopy and Pathology decided that this biomarker was validated for its use to diagnose cervical biopsies. And then in 2014, the World Health Organization followed with their guideline recommendations supporting use of this biomarker. But to the point that you made, for every one biomarker that's effective like this, there are thousands that don't do what they're intended to do. So it's a very difficult exercise. So as I mentioned, we need to take any idea that we have like this. We can't just put it on the market and say, believe us, this helps cervical biopsy diagnosis. We have to do a study that's going to convince the FDA that this is a safe and efficacious product. So in this case, we did a very large study called the CERTAIN study, that stands for Cervical Tissue Adjunctive Analysis Study. It involved 70 pathologists, 1,100 biopsies, and 38,000 different slide reads. So a lot of data. So this is the level of study that's required to convince the FDA that the product is safe and effective. I'm not going to go through all the gory details of the study, but basically we did what you would imagine we would do. We compared pathologist analysis of cervical biopsies with and without our test. So we had them look at the same cases with a washout period in between with just H&E, so the old way, and then gave them our test with the H&E, and then measured how accurate they were compared to experts, which is over here. So this is the data. Who's familiar with ROC curves? Okay. So ROC curves is a way of looking at sensitivity and specificity. So obviously you would want 100% sensitivity, so sensitivity, the ability to detect true positives, and specificity, the ability to detect true negatives. So the way this is plotted, perfect sensitivity and specificity is up here. So this is the plot of all the 70 pathologists, so one dot is one pathologist. This is their performance H&E only. So you can see this particular pathologist, right? So they had roughly 50-something% sensitivity, but close to 100% specificity. So what happened? So watch the dots. So you can see if we look at this pathologist, with our test, they improved from about 80 and 80 to 90 and 95, right? So that one pathologist improved both sensitivity and specificity. Now, if this will work, you can see how the performance of all the pathologists did. So you can see the vast majority of the pathologists improved sensitivity and specificity. There were a couple of pathologists who you saw going in the wrong direction, but the majority then did improve. So here's before, their sensitivity and specificity before with the H&E only, and here's their performance after. So based on this data, the FDA was convinced that if we provided this to the world's pathologists, that we would be doing more good than harm, which is how the FDA looked at it. So in March of 2017, the FDA issued technically a clearance for this, and that gets into some regulatory detail that you don't need to be concerned with. But basically, we were granted approval to market and commercialize this product with the claim around improving diagnostic accuracy for cervical biopsy diagnosis. Okay, so here's the quiz. So I'm going to demonstrate live the power of this test. So I have 3 examples. So here's the H&E, and I want to ask you, what is your diagnosis? Do you think it's benign or cervical? So in a second, I'm going to ask you, so you think it's benign, I want you to hold up one finger. If you think it's cervical precancer, some form of CIN, I want you to hold up 2 fingers. Okay? So I'm going to count to 3. I'll give you a second to look at it. All right, here we go. 1, 2, 3. Higher. Okay, look around. Right? So you see some 1s and some 2s, right? I'm not going to count them, but I think maybe slightly in favor of 1. Okay? But a good mixture. I would say 2/3, 1/3, something like that. And now I'm going to show you the test, and I'm going to ask you again. Okay, so here's the result of the test, brown staining. So now I'm going to ask you again. Is it benign or cervical precancer? 1 finger or 2? 1, 2, 3. Now look around. Everyone's holding up 2 fingers, right? So that's the power of the test. So you just proved it. So you started out 2/3, 1/3, and then you went to virtually 100%. So that's exactly how this test is intended to be used. Here's another example. Okay, I'll help you a little bit. What's your diagnosis benign or cervical? Here's a cervical gland. This particular biopsy has a lot of inflammation around it, which can cause cells to look funny. So based on that, what is your diagnosis? 1, benign, or 2, precancer? 1, 2, 3. Okay, now I see mainly 1s. I see a couple 2s. I think I biased you. Okay. Now here's the test result. So is the test negative or positive? Yeah. Oh, right. I should have said that before. It needs to be completely. You need to see a broad streak of brown. Like 1 or 2 brown doesn't count. So this is negative. This is negative. Okay. So knowing that the test is negative, now give me your diagnosis. 1 or 2. 1, 2, 3. Okay. Now everyone is 1. Okay, we'll do one more. Okay. Here's another biopsy. I won't biopsy, I won't say anything. So here's the H&E, your diagnosis is benign or precancer. 1, 2, 3. Twos. Okay, a lot of twos. A couple ones, but mainly twos. Okay? So you think it's precancer. Pretty good. Pretty good. So you get the idea, right? So you can see the power of this test. And so I'm happy to say that not only is this product doing well commercially, but with the feedback from the field of pathologists is it couldn't be better. That people really appreciate having this tool. Not that they always use it, but especially for the difficult cases, this really helps them figure out what decision to make. When I was a resident, actually a fellow, I did a fellowship in gynecologic pathology, and believe it or not, when we would have sign-outs, meaning the pathologist would sit with all the residents and fellows around the scope. Sometimes, our lead pathologist, the teacher, didn't know what it was, and so she would take a vote, and so we would literally vote on just like we're doing now, benign, and the majority won. And who knows how many times we got it right or wrong. That was the old way. This is much more objective. I think you would agree. So these are the people. So these are the people that work in Oro Valley that developed that test. So it's a relatively small team, and the majority of these people are medical, scientific, clinical people. So this is the type of work that you can accomplish and the impact that you can have around the world by applying medical and scientific principles in an industry setting. So I think we have time for the, or actually maybe not, maybe
5 minutes.
5 minutes? Okay.
Well, at least 1 or 2 minutes.
Okay.
A few minutes.
I'll just really very quickly touch on this second example. And who's heard of cancer immunotherapy? A couple of people. So if you see the graphics here, so Jimmy Carter thinks that and he's correct, that he was cured of his disease through his brain cancer through immunotherapy. So this has made a huge impact to the field of oncology. This is a completely new way to think about treating cancer. And I just want to show you a little bit of the excitement and some of what Ventana is doing here. So this is a PET scan of a patient with melanoma, and all the black dots is metastatic disease. And this is a repeat PET scan, if you can believe it, 4 months later. So you can see the vast majority of those black dots have gone away, and that's due to this immune therapy. So this really works. This is a bit of a gruesome photo. This is, again, melanoma coming through the skin. Widely metastatic. You can see over the course of 2 years, completely going away. And you'll notice that between here and here, it actually gets bigger. This is because the immune therapies, what these therapies do is inflame the tumor, get immune cells to go in there, and the immune cells kill the tumor. What's impressive about these therapies is that patients are surviving. This is 10-year survival data. So we have patients who started with metastatic disease, and at least 20% of them are alive at 10 years with no evidence of disease. So this has never happened before in cancer therapy. The way we think this is happening is that cancer cells spill some of their proteins into the body, then cells pick up those proteins, present them to the immune cells, activate those immune cells, and then those immune cells, specifically T lymphocytes, go back to the tumor and kill them. So I'm going to show you, just because we have a very little time, this is a T cell, the green cell, attacking a tumor cell, which is in blue. So you can see how aggressively that, almost like the blob, spreading out across that tumor cell and literally digesting it. So who knew all along our immune systems and all our bodies are perfectly capable of killing cancer? I'm going to stop the video in the interest of time. The problem is, so if our immune systems can kill cancer, why does cancer even exist, you might ask? The problem is this, that also, as you could imagine, through the millions of years, hundreds of millions of years of human evolution, we needed to regulate the immune system very carefully. Because if we didn't, then every time we got a cold or flooded infection, our immune system would stay activated and kill us because it would attack our normal tissues. So the immune system has responded to that by creating on-off switches, regulation. So these are all the different switches. So the bottom line, it's a very complicated regulation system. Unfortunately, tumors have figured out a way to take advantage of that regulation system and shut off the immune system. And one of the shut-off switches is this PD-L1. So tumors can express PD-L1. The T cells have the receptor for PD-L1 called PD-1, and then this interaction shuts off the immune response, essentially. So the idea is, can we develop drugs that interfere with that off switch activation? So if you prevent the off switch from being activated, then you essentially turn on the immune system. That's the idea. And so we have a number of drugs that do that. One of them is called Tecentriq. And what's interesting is the tumor is not targeted. You're targeting the immune system. You're not actually targeting the tumor cells. You're allowing the immune system to do its job to kill the tumor cells and fight them. I'm going to skip that. I'm going to skip this. Oh, well, basically, this PD-L1 mechanism is expressed in many different tumor types, and these drugs have shown efficacy in almost all of those tumor types. And you can see the response rates here, very high response rates. And now to get to the testing part. So Ventana has developed a test for PD-L1 that helps predict which patients will respond to these therapies because they all don't respond. And so now the brown before was P16, the brown in this test is PD-L1. So when a patient is positive for PD-L1, they are more likely to respond to these immune therapies. That's the bottom line. And in the interest of time, maybe I should leave it there. But this shows the different tests. So these are the two Ventana tests. The other two are from our competitor called DAPO. These are the 4 PD-L1 tests that are approved and on market for these immune therapies. But it's an extremely exciting field, as you can see, and we're just in the early stages. So if I ever come back, hopefully I'll have time to tell you the rest of the story. But this could be the cure for cancer, immune therapy. And up in Oro Valley, we're working on a part of it that involves testing of the tumors for this PD-L1 marker. So why don't I leave it there and maybe go to my summary slide. And just wanted to hopefully leave you with 3 things. The first is, like I said in the beginning, I encourage you to have goals for your career and pursue them aggressively, but realize that your ultimate path may be different than the one you have in mind. Just be open to that. The second thing that I would tell you is that you can absolutely have a fulfilling and rewarding career in industry by pursuing science and medicine, and along the way, you can benefit the lives of cancer patients around the world, like I feel I have. And third, follow your bliss. So thank you very much for your time. I'm happy to take any questions, or if you'd like to come up afterwards, I can stay. Thank you.